Welcome back to the castle. Today we're going to look at part 3 of this series on the science of precision irrigation, why it's so important to your crop's operational success, your return on investment. We looked in the past 2 videos at some of the physical characteristics of soil, which we'll look at in a second, and some of the basic properties of water. In this video, we're going to look at the mechanisms that dictate how water moves both through soil, into plants, up through plants, and back into the atmosphere. There's going to be some science in this video. And some of you may be saying, oh, here we go with the science. However, with all the videos that I've done in the past, this video has the potential to tie in all of the other subjects we've talked about in the past. Soil structure, soil temperature, irrigation levels, humidity, vapor pressure deficit, light intensity. You've heard me talk a lot in the past, especially if I've talked to you in person, about this principle called the law of limiting factors, that in a grow operation, a grow room, grow chamber, greenhouse, that there are many different things that have to be in perfect sync with each other for the success of your plants. Again, things like temperature, humidity, light intensity, duration of light, irrigation levels. If we have, say, 8 or 10 of these different elements that have to be perfectly synced for the best quality and the highest productivity, if one of those factors is suboptimal, the entire grow operation can only operate at the speed allowed by that one suboptimal element. So we're wasting time and money if all of them aren't optimal. But a lot of people, most people, don't understand how these things are all linked. This video today, when we talk about irrigation science and water movement, will bring together why all of these things are so closely related and how minor variations in one property or one element in this continuum, how that can negatively affect your entire grow. So the first thing I want to do is just take a 1 or 2 minute review of some of the things we looked at in the past 2 videos, and then let's dive into the science. And again, I think the science on this is fascinating, and for a lot of you who are serious growers and who want to understand how your plants function, how the different elements in the environment affect each other element, I think you'll find this to be a video that is going to clear up a lot of the confusion of how my plants grow and why we need to be such good stewards of all of the environmental levels that we're giving to our plants. So let's start the process. In the past 2 videos, we looked at soil and soilless medium. We looked at the composition of soil. What is soil? We looked at that combination of elements like sand, silt, and clay, the different particle sizes. We looked at why the different particle sizes make such a difference to how that soil functions, mainly dictated by the size of the pores. If we look at the different types of soil, we have very coarse particles, we have very fine particles. The coarse particles give aeration and drainage. The very fine particles are responsible more for holding on to water. We need both of them in a soil that functions optimally for the crops we're growing. And then we looked at water and looked at basic water molecules. We have an oxygen and 2 hydrogens, we know that we have a slight shift of the electrons towards the oxygen molecule, which gives us a slight positive charge on the hydrogen side and a slight negative side on the oxygen side. This is what gives us this dipolar element of a water molecule. This is going to be very critical when we start looking at how water moves through soil into and up through plants. We also looked at, in addition to having sand, silt, and clay in our soil or growing mixture, we also have organic materials. We know that these organic materials can do what we call aggregate and form kind of clumps that have organic compounds in them, have different chemical surface charges, but in general, our soil, between the organic and the mineral components, the soil carries a slight negative charge overall. Again, this is going to be critical as we start looking at the movement of water through soil into and through the plant. We delved a little further into the properties of water molecules and how important this dipolar, this positive-negative aspect of a water molecule, how that affects water's movement. We looked at 2 critical things that happen with water. We looked at cohesion, which is the sticking of water molecules to each other through hydrogen bonds, cohesion. So we get them traveling together if they're being pulled in one way or the other, either by a pressure component or by gravity. We look at the properties of adhesion, which is the sticking of a water molecule or water molecules to non-water surfaces. It can be the mirror, the piece of glass that we looked at in the last video, but in the case of our water and our irrigation science, we're looking at the sticking of water molecules to the surface of the particles in the soils, be they the mineral components or the organic components. So we've got cohesion, we've got adhesion. These, again, are going to be critical as we start looking at the movement of water. When we look at these 2 properties of cohesion and adhesion, we then looked at the principle of capillarity, or the ability of water molecules to rise in a narrow tube. If you stick a narrow tube like a pipette into water, you'll actually see a traveling of water up into that pipette. That is due to those adhesive forces, that attraction of the water molecules to the glass, in this case of the pipette. It will have a tendency of drawing that water up, but it'll only draw it up to the point where the opposing gravitational force balances it out, so that the pull upward is balanced by the pull downward, so we get a particular point at which that water will rise to. The wider the tube, the lower that level, because the gravitational forces become stronger. So when we're looking at the movement of water into and upwards through plants, this capillarity, this traveling up a narrow tube through a combination of adhesion and cohesion. The adhesion is having the molecules be drawn up by that negative charge on the glass. The cohesion is, as the top molecules are pulled up, you have this sticking of the water molecules behind it, pulling that column of water up again until you have a balance between the upward pull and the downward pull of gravity. At the top of that water column, we will have the formation of of the meniscus. And those of you who are familiar with your high school chemistry, it's that kind of concave structure at the top that is the boundary between the air and the top of that water column. So we have the meniscus at the top, we have the adhesion, cohesion, pulling that water column up, and again, this function is going to be critical in understanding irrigation and movement of water up and through the plant. The meniscus at the top is going to be where a lot of the action happens, both in the soil and in the leaves, as we look at the process of transpiration, where the water in the plant being drawn up is then released into the atmosphere. So cohesion, adhesion, sticking to some other surface, and then we have capillarity, or capillary action of that movement up through a capillary tube and the formation of a meniscus on the top. So now, let's see if we can put this science together and see how all of these different properties start functioning and help us determine how and when to irrigate our plants. So, the first concept we really have to look at, and that will form the foundation of everything we talk about in irrigation science, and again, all of your set points in your grow chamber. This is going to be the concept of water potential or water energy potential. And we could spend a lot of time on this, looking at the physics, and it could make it really complicated. But take a breath. This is going to be really easy to understand, and it's going to form the foundation of your increased knowledge of how plants function and how to run your grow operation. Water potential is a way that we quantify the energy of water in a particular place and state. If we have water sitting here, and by water we're saying pure water, being distilled water, RO water, water that in theory is just H2O molecules, nothing else in the water. So we have water sitting here, pure water, in a glass container, and we can call this the reference point. When we talk plants, the reference water is going to be pure water at the surface of the soil level in our grow medium or our garden. So first thing, water potential, pure water, we need a reference, and it's going to be at the level of the soil surface. The next thing we want to look at is what is a different energy level than this reference sitting here. If I've got another container of water, let me zoom in here a second. So if we have our reference sample, and we're at soil surface level, and I have a second container of pure water, and I set them side by side, and they're at the same level, they both have the same energy, the same water potential. The easiest way to change the potential between these two, I lift one container up. From a gravitational standpoint now, we have changed the energy, the water potential here. A very common example of how this works in everyday life, we just finished doing a video of the water supply, the water purification plant in Rochester, and they were talking about how 80% of the water in the city is delivered through gravitational means. They build a tank high on a hill. They pump from the purification plant up into that tank, and when you open your tap, water will flow from the high point down to your tap. The reason we put the big tank on the hill is because we want to increase the water potential. We want to increase the energy in that water. This defines probably the key thing we're going to talk about in this whole video. If I raise one up, and I've got a clear tube that connects here to here, and there's a little valve in it. When the valve is closed, because the water can't go anywhere, these two systems will remain exactly as they are. If I open that valve, the water is not going to flow from the lower glass into the upper, and you may think, well, that's obvious. It's going to flow from the upper to the lower, but this is an absolute critical principle that we're going to look at more in depth, that water wants to move from a situation of higher water energy to lower water potential energy. Very simple. How's that apply to us when we start talking soils and grow medium? If I have a grow medium here, this is the very coarse sand, and we did this, the large gravel, just as an example. If I pour water into here, the water is not going to sit on the top of the gravel. It's going to do what? It's going to run down through, because the water at the surface, as we pour it in, has a higher potential energy than the water down here, because there really is no water down here. So it's going to go from the area of higher water energy, water potential, to an area of lower. So this is the number one easiest to understand element of water potential, the gravitational component. And again, I've increased the water potential, the potential energy of this glass just because it's higher than this one. So again, think of it as your soil. Water wants to move from an area of higher energy potential to an area of lower potential, whether it's in your garden soil, your farm field, or in the grow medium in your greenhouse or your grow operation. So water energy potential. But water energy potential is affected by more than just gravity. Other factors that can impact water potential energy, either in a positive or negative way. We spoke about gravity, the gravitational component. We can also have a pressure component. The next element of understanding water potential when we start talking soils and plants, you're familiar with the term, and this is probably the second most important element of water energy potential that we will consider as we look at, again, the movement of water through soil, and especially into and up through the plant. This component is the osmosis, or the osmotic potential component. For those of you who remember some of your high school chemistry or your plant biology, osmosis is a process that takes place in virtually every living organism. We use it in things like water purification, production of chemicals. It's a very ubiquitous occurrence in the real world. So for those of you who aren't familiar with what osmosis is. Again, take a breath. This is going to be easy to understand. We may upset some of the physicists and the PhD biologists because we're going to bring it down and just leave some of the things out. But what the principle of osmosis is, it hinges again on this concept of water potential. If I have two containers here, two containers of pure water, forget that this one's blue at the moment. We'll, again, this is just for coloration. So at the moment, these would be two very identical containers of water, the same height, so they've got the same gravitational potential. They're both pure water, just H2O molecules side by side. They will not really react in any other way outside of if we cut the wall between the two. They would just mingle and become one container because they both have the same water potential energy at the moment. However, if we take one of those containers, which we've already done, and we put what we call a solute into it, and if you watch any of my fertilizer videos, you know that all of the fertilizers that we use are water soluble. When you put them into water, usually the granular fertilizers like this particular fertilizer here, they will then dissolve into their ionic components. So they're adding things like magnesium ions, calcium ions, sodium ions, chloride ions. So we just have this soup now of H2O molecules with additional molecules of non-water components. Again, our mineral salts, calcium, phosphorus, magnesium, yada, 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 which we have done in this blue container here. Now, a thing to understand is we started with pure water, just H2O, strictly H2O molecules in it. When we put a solute in and it dissolves, what happens is the water molecules now have a tendency of kind of gathering around, and they have some ionic attraction to these molecules, which does what? It lowers the energy potential of that water. When we put a solute into water, it lowers the energy potential of the water because the water molecules kind of come occupied with being attracted to those solute molecules, so the water potential drops. So that if we put these two side by side, or if we put them into one container that has a membrane between this side with the solute and the side without the solute, if we put a piece of glass between the two that can be sealed, nothing will happen because we've blocked off the path. But we have to put what we call a semi-permeable membrane between the two containers of water. A semi-permeable membrane is one that will allow water molecules to pass through, but will not allow the solute ions to pass through. Semi-permeable. You might say, when am I ever going to see that? Basically, every cell in your body and every cell of your plant crop, the cell walls are semi-permeable membranes. This is going to be critical in our understanding of movement of water up into and through plants. So we have pure water, a water with solute in it, separated by a semi-permeable membrane. Remember, water wants to move from an area of higher potential energy to an area of lower potential energy. When we put the solute in, which in the plant world is usually our fertilizer, when we put the solute into the water and put a semi-permeable membrane, lower water potential energy, higher water potential energy. What happens? The water is going to, molecules are going to pass through the semi-permeable membrane from the container of higher water potential to the container of lower water potential. If we look at a cell wall or the outer layer of a root hair, these are semi-permeable membranes. So in a cell, the cell, a lot of what it tries to do is to regulate how much water comes in, how much stays out. We have active transport channels that will allow it to take some of the solute molecules in the fertilizers when it needs to, but block them when it doesn't. But the basic principle is that the inside of the cell is separated from the liquid in the plant up through the xylem by semi-permeable membranes, and that the cell can control how much comes in and out through that semi-permeable membrane. You might be thinking, yeah, well, why is that very important to me? If we look at a plant in your grow operation, and I see this all of the time, and I'll go in and I'll ask people when they water. Well, my buddy told me to do it when it looks like this, and my buddy told me to do it when it looked like this. A common answer that I hear is, I water when I see the first signs of wilting, which makes the hair on the back of my neck stand up. If we have a cell, we're going to look at this balloon as a cell. So we have a cell. The outside of the balloon is a semi-permeable membrane. Inside the cell we have the nucleus, the mitochondria, all sorts of other little components of the cell which allow it to operate efficiently in a biological sense. Out around the cell, every cell is in some type of a contact with water being transported by the plant to be able to bathe that cell both in water and in nutrients and in sugars and in starches. And the cell wall can control how that gets in and out. So you might be looking at this and say, well, what does this osmosis have to do with my irrigation? If we have plenty of water in the plant, which we're going to talk about how it gets there, and you have water surrounding the plant, you have nutrient solutions surrounding the plant, the cell reaches a point where it's happy. I'm going to put a little more cell water in there. This cell, by controlling the osmosis, builds up pressure in the cell wall, because the cell will actually produce compounds inside the cell which will make the water potential of the water inside the cell lower, because it adds things like starches and produces sugars that lower the energy component, so that the higher energy water on the outside of the cell actually travels into the cell. When it travels into the cell, it actually kind of bumps up against the inside wall of the balloon, being the cell, so it makes the balloon rigid. I mean, that's why it looks like a balloon. If we don't have enough water being supplied to that plant through irrigation, so that the plant can't bring water up, and the amount of water outside the cell starts to drop, and the osmotic potential lessens, what happens is the water inside the cell is actually drawn now out of the cell, which I'm doing by letting the air out, and you're going to get to a point where we have wilt. This is what's happening in wilt in a plant. When the cell is fully hydrated, it's in a state of what we call turgor. That's when the leaves are standing up nice and they're nice and folded out, and the plant looks good and healthy. As soon as you start seeing wilt in that plant, it's an indicator that the plant cells no longer have the water available to bring into the cell to increase that pressure in the cell to get that turgor. If you're watering at that point of wilt, you're way past the point where the plant is suffering various forms of stress, and you're actually slowing down the production and costing yourself time and money, and you're costing quality and potentially the health of the plant. If you get to a point where this is so wilted, it's a point we call the permanent wilting point, where even if you start to water, the plant now has passed that point where it can even bring the water up. So we're going to look at this osmotic potential as critical in our process of keeping our plants fully hydrated. The water is going to carry the nutrients, it's going to carry the sugars, it's going to carry the starches, it's going to be responsible for cooling the plant. Once we don't have enough water available to that, all of these processes become suboptimal, and the plant is doing nothing but suffering and costing you quality, productivity, and return on your investment. So osmosis. Most of you who have talked to me in person know how much of a stickler I am for using an EC meter whenever you're making up your fertilizer batches, so that you know what the EC of the water going in is, but how much I'm also an advocate of doing outflow EC readings. The reason being that we just talked about how water wants to move from an area of higher water potential into an area of lower water potential. We put a solute in, and pure water will want to travel into the side with the solute. If we look at a root hair and we have a grow medium, and we take this fertilizer, saturate our soil with the plant in it, when you think of how that fertilizer and that water is going to get into the soil, now we're putting in something that has a solute into it in the form of a fertilizer salt. So this has lower potential energy than the pure water. So how does it get into the root? Because osmosis is one of the ways that water has to get into a root. But we already have a solute in this. So the obvious answer is that on the other side of that root hair, and the outside of a root hair is a semi-permeable membrane, on the other side, which is the inside side of the root, there has to be some lower potential energy than as is in this solute. So how does the plant make sure that there's a lower water potential inside that root than the outside, so that the water and carrying some of the nutrients can flow into the root instead of going out? We mentioned that when we were talking about individual cells, that the plant can actually produce starches and sugars and other compounds that it will put into the fluid inside of its own vascular system. And in this case, we're talking about the xylem, which takes water from the roots and carries it up through. You're probably familiar with phloem. That's the channel system that actually moves around the plant and kind of has a downward flow and a lateral flow. So how does the water, which is already has a solute and a lower water potential, get through a semi-permeable membrane into the root? Obviously the root, there's another component we'll look at shortly, but on the osmotic end, the root has to produce a slightly higher level of solute on the inside of the root so that the water is still going to flow in this direction. The plant can do this quite easily if everything is functioning well in its biological system inside, and we've got all of the components of the light intensity, the irrigation levels, the temperature. But if we make this fertilizer solution, this solute solution, too hot, and by too hot we mean that EC is too high, we may get it, very well get it, and I see this a lot in grow operations, we may get this solute solution so high that the plant cannot really counterbalance with a higher solute solution inside, so that you get a dramatic slowing of the plant's ability to pull water and nutrients in, and we might even get it to the point where the solute in the soil out here and the water is so high that, say, in a young seedling, it's actually pulling water out of the seedling, out of the roots, and it actually kills the seedling in the process called plasmolysis, where you actually suck the water out of the seedling. So this is why, from a grower's standpoint, it's so critical to control the EC in the nutrient level going in, and to also know what that outflow EC is, which is going to tell you, are you getting a salt buildup in the soil outside of the root? If that salt buildup is going up and up and up and up and up, you may get it so high again that this plant is fighting to get any water in, because it can't counteract the differential in the water potentials, because the solute outside is actually much lower water potential than the solute inside. So this stuff becomes critical. We're going to see how all of the components of the grow operation above the soil is going to even affect what's going on that we're talking about here with the osmosis in the root here. So osmosis, critically important, has to have a semi-permeable membrane. Just like gravity, water wants to flow from a level of higher water potential to a level of lower potential to try to stabilize. Put a solute in, lowers the water potential, so water molecules will travel from the area of higher potential to the area lower potential. If there's a solute in both solutions, on the inside and the outside of the cell wall of the root here, if you want water to come in, the osmotic potential, that water potential, has to be lower on the inside of the root. So this, again, is why we have to pay critical attention to our fertilizer, our solute input, and the outflow. Now we're going to take a look at the number one prime mover of water in this soil, root, plant, air continuum of this movement of water from the soil into the plant, up through the plant, back out into the air. This component of water energy potential is what's known as the matrix potential, or the soil matrix potential. We touched on it in the last video when we looked at the fact that soil is made up of different grains. This is the very coarse gravel to be able to show you the grain size, and then we went down to the finest being clay and silt, which is like powder. We looked at the fact that with a large coarse granule, we have much bigger pores, but we have much less surface area on those pores. We have bigger pores, which are called macropores. If we put water in, these macropores are so big and the gravitational component is so high that you get immediate, very heavy flushing. The only water that's really held in these is when you have grains touching, and we bring in this capillary component of the water molecules actually adhering to the surface of these granules and forming these columns where they have a meniscus on the top. So the larger the grain size, the higher the gravitational component, and the lower the water retention. When we look at this matrix potential, the matrix potential originally was called a capillary suction potential, soil suction, because it was assumed that the capillary component, where you have grains that are very close together, so we get that pipette action of the adhesion, drawing those molecules up into those spaces between. But then research showed that actually the biggest component of this matrix potential is actually the adhesion, that attraction of water molecules actually to the surface of the particles in that soil. And remember, in general, the particles of the soil have a negative charge, and that gives a corresponding positive attraction of the water molecules. So you have this adhesive force, the sticking of water molecules to the surface grains. And if we look at this diagram that we're looking at now, is probably the most, that's probably the second most important diagram we're going to look at in this video. As we can see in the diagram, we have a soil particle over on the left-hand side, and we're showing the water molecules in that space to the right, which is an air space in the soil, a macropore. You've got the cohesion of the water molecules in that macropore, and then as you get closer to that particle grain in the soil, you start having this adhesion, this molecular attraction of the water molecules to the surface of that particle. And these forces are much stronger than the cohesive forces out in the middle of that macropore. So if you look at the gradient, and we're always going to be talking here about energy gradients or potential gradients, out in the middle of this macropore, where you just have cohesion holding the water molecules together, and we have a root hair out there. The root doesn't have to work very hard to get those molecules, those water molecules, just to break apart from each other from the cohesion they have with each other. And by osmosis and one other factor we're going to look at shortly, it can start to move those across that semi-permeable membrane of the root hair into the root and start working them up the plant. But the more water we draw out of that macropore, and the closer the water gets to the surface of that grain within the sand, the higher those adhesive forces between the water molecule and the surface of that soil particle gets. The higher that gets, the more energy is taken from the water, which means the energy potential of the water is lower and lower and lower and lower as we move from the center of that macropore to the surface of the soil particle. Now we measure that, whatever you want to call it, the suction, the attraction, that matrix potential, that draw, those adhesive forces. we can actually quantify how strong those are. And we can measure it in a number of different units. The two most common would be like the centibar, the bar, kilopascals, megapascals. Don't even worry about what those mean, how they relate to each other, what the conversion factors are. Just understand one simple concept, that reference water at the surface area of the soil will always be referred to with a water energy potential of 0. So we're going to use the reference of 0. As we irrigate and the plant starts pulling water out of the soil, and we start having these adhesive forces from the particles start affecting that water and having that attractive force, which drops the energy of the water. And we do things like put fertilizer in, which again adds more or drops that energy potential more. As we take more water out and the attractive forces get higher and higher, the energy level gets lower and lower and lower. So when we quantify that, we always use a negative number. So if we're using something like a kilopascal, a water that's almost saturated, a soil that's almost saturated, let's say it's got a kilopascal rating of -10. You take a little more water out so that there's more attraction from the soil particles, we're at -30, 40, 50. We take more out, we're down, we can measure it as a holding capability, -100. We can get down to -500, 600, 700, 800. So the less water in the soil, the drier the soil, and the more solute in the soil, the lower that number will be. And the harder that plant root and that plant has to work to get water out of that soil. It has to create more osmotic differential by creating more sugars and starches in its root to try to suck harder with the osmotic potential, and it's going to do a couple other things we're going to talk about in a minute. So the key element here is to understand that as the water level in the soil drops, it becomes harder and harder and harder and harder for the plant to get water out. And as we're going to see in the next video, the point at which it becomes very difficult for the plant to get water out and where the plant is actually experiencing quite a bit of stress is much higher in terms of the amount of water in the soil than the average grower thinks is necessary. So the water becomes harder to pull out, the less water there is in the soil, and the more solute there is, fertilizer there is in that grow medium or that soil. Again, don't worry. You don't have to know kilopascal, megapascal, centibar, bars. Just understand that 0 is the reference. Negative means that the water energy potential is lower. The lower that negative, -5 is lower than -1. -10 is lower. -100 is much lower. -500 is much lower. That all relates to that holding power of the soil for the water in it. And the lower that number, the more negative that number, the much more difficult it is for the plant to get that water out. And we're going to look at how to put all of this together now by looking at probably the number one diagram that we're going to consider in this whole discussion. So this graphic that I'm putting up now is probably one of the best graphics I've seen, and probably my favorite graphic to help people understand this whole concept of water potential, of movement of water from the soil into the root hair, into the plant xylem, up through the plant, and then out into the atmosphere. Again, this soil, plant, air continuum. One of the reasons I like it so much is that it will now bring together all of the things we've been talking about in the past 3 videos. And if we spend a little time on it and you just relax, and there's nothing really complicated here. We've already covered most of it. Now we're just going to kind of put it all together. So let's start from the bottom of this graphic, down in the bottom right, in the root zone. We have the roots of this tree in the soil, and you'll notice this number that says soil, negative 0.3 megapascals. I actually think that's supposed to be 0.03 megapascals. And just remember, don't worry about megapascals, bars, centibars, kilopascals. Just understand that that is a number referencing the that potential energy of the water is in that soil. It's a negative number, so we know that that is lower than that reference water, that pure water at the soil surface of 0. So we know we're starting with a negative number, which means there is some holding of water in the soil by those soil particles and the effect of gravity and the attraction to organic compounds in the soil. If we look at the little diagram, the square on the right, number 3, that's basically a little illustration of what's going on. We can see the root here protruding out into the soil. We see the soil particles, and the blue around the soil particles being the water. And we can see water molecules moving into the root hairs. Now remember, one of the primary drivers of the water molecules coming into the root hairs is this osmotic potential. The outside of the root hair is a semi-permeable membrane. The root has produced starches and sugars that give a higher solute level on the inside of the root than the outside, so that the water is going to move from the area of higher energy on the outside of the root hair to a lower energy state on the inside, because the solute level in the root hair is higher. So now we talked about this movement of water from a level of higher energy to lower in osmosis, but the process of osmosis isn't strong enough to get those water molecules up to the top of that, whether it's a 10-foot tree or 50-foot tree or a 100-foot tree. How the heck is that water getting up to the top of the tree to feed all of the cells, to cool the plant, and then leave the plant? So we see the water has come from the soil through that semi-permeable membrane of the root hairs into the root. But now, how are those water molecules getting to the top of that 100-foot tree, or your tomato plant, or your medicinal crop? How the heck is it getting there? Because there's not enough osmotic draw differential between the water energy in the outside the root here, and inside, to drive that to the top of that 100-foot tree. So what's going on here? Now we get into the meat and potatoes of how this system works and how we have to manage this system. If you look at, on the left-hand side, that blue arrow that is pointing up from the soil to the top of the tree, and you look and see what it says, water potential gradient. Everything we've been talking about today is wrapped around this concept of differences in water energy levels, which is also called water energy gradients. There's some type of a difference between the water energy gradients going on in this plant, because the only way water is really going to travel is to go from a level of higher energy to lower energy. So what is driving this? If we look at the very top of the tree, we can see that the outside air, the water energy potential of the outside air is negative 100 megapascals. Again, don't worry about the megapascals. Just worry about the fact that the top of the tree, the outside dry air, can have a level of negative 100. Remember, down at the root, we were looking at a level of negative 0.3, or actually 0.03. So there's a huge difference between the two here. And what direction is water going to travel? Water is going to travel from a level of higher energy to lower energy. Negative 100 is a lot lower than negative 0.03 or negative 3. So we've already established that there's a huge water gradient difference going on between the soil and the outside air, because the outside air is very dry. So we have a tendency of the water wanting to move in that direction. But how is it going to get there? If you look again at that number 3 box on the right, we see the water coming in to the root here, and then what it does, it travels into the tubing, the vascular system of the plant. In this instance, the xylem, which is those tubes that carry it upwards into the plant. And remember, when we were looking at capillary action, if I take a very narrow pipette and stick it in water, water in that pipette is going to travel up inside that tube by the adhesive forces drawing the water up, and the water molecules sticking together in cohesion, so it pulls the water column up. But again, it won't pull it all the way up the tree just by capillary action. So how does it get to the top of the tree? If we look at box number 1, the very top of the xylem terminates, and what happens at the top of that termination? You get an area where the top of the xylem and the cell walls are actually exposed to air in the leaves. And remember, that portal for air, you're all familiar, if you're growers, with the stomata on the bottom of the leaves. They're actual openings in the leaf that the leaf can open or close to let carbon dioxide in, oxygen out, and let water vapor in and out. So you have this opening to the outside air, this opening to this air with a water energy potential of negative 100. Some of that dry air with that negative 100 factor actually comes in contact with the meniscus, that curved structure at the top of a water column. It comes into contact with that water at the top of the xylem that kind of gets the cell walls wet. And what happens is that the energy of the sun, the warmth, and the dry air actually cause the water molecules at the top of that meniscus. It causes the hydrogen bonds that have the cohesive action in water. It causes those to break by adding the energy of the sunlight and the warmth. It causes those bonds to break. Those water molecules turn into a gaseous state. They exit through the leaf. And what happens when those leave the top of the meniscus, the surface tension at the top of the meniscus and the cohesion brings water molecules up behind. So as you break apart the bonds and you have the water vapor in the form of gas leaving the top of the meniscus, the tension of the cohesion actually pulls more water molecules up behind. So you have leaving and then pulling up and leaving and pulling up and leaving. So you have this tension, and this system is actually called this transport system, and this ability to pull water up from the roots to the top of the plant is called the cohesion tension system. The tension is the pull of the dry air, because the dry air is at -100 as opposed to the 0.03 at the root. And if you look at the different levels of energy up that water potential gradient, at the root, they've got it as -0.3, which is a little higher than at the trunk, which is -0.5, which is a little higher than the trunk system, which is -0.8, which is a little higher than the leaf at -1, which is a little higher than the leaf air spaces at -7, which is a lot lower than outside air at -100. So this is the absolute key. We've been talking about water energy, water potential gradients all through this video. Water wants to travel from a level of higher, and 0.3 or 0.03 is much higher. It's higher than -0.5, higher than 0.8. So water wants to go in that direction. And the 100, the -100 is a strong pull. And the xylem tubes are so small, and the cohesive action of the cohesion of the water, keeping the water molecules together, and the adhesion of the water kind of attaching to the walls of the xylem, and the pull of that -100 dry air outside at the evaporation point of the meniscus, that's what drives the water. So you've got at the root water entering from the soil into the root, partially through osmosis, but then taken over by this process of water wanting to follow the pressure gradient to go from higher pressure water potential at the root to a much lower at the leaf interface. Again, whether we're growing lettuce, tomatoes, peppers, blueberry bushes, or 100-foot high sequoia trees, it's driven by this cohesion tension. So now as growers, you can get a little bit better idea of all of these things that you have to be sure to take into consideration when you're operating your grow room. The number, probably a couple of the really important ones are how much water is available to the roots of your plant. Because remember, a lot of you seem to water when you see the first signs of wilt. My buddy told me to wait until it's wilting and then it's all right. If the plant is wilting, it's not transpiring. It's not doing this process of pulling water up and evaporating it in the leaves. The plant is not able to cool itself. It's not able to pull water and nutrients up. It's not able to move nutrients and water around the plant. So the plant is basically sitting there and being like, we'd like to be growing, but we can't really. We don't have the water to be able to manage and operate our biological systems. So in the next video, we're going to look at how you take all of these figures and apply them to a very precision-driven irrigation system. But if you trying to figure out how to manage your lighting system, the intensity of the lights will dramatically affect the rate of photosynthesis, in the plant, which will dramatically increase the needs, the water and nutrient needs of the plant, so that if you increase your lighting intensity, you have to make commensurate increases in the availability, the continual availability of water in the root zone at an appropriate water potential level. Most growers I see drop below that optimal level all the time, and they realize lower than optimal growth. So your light intensity, an aspect of grow room operations that people don't pay much attention to, that's critically important, is the whole concept of vapor pressure deficit, the relation between the humidity in your grow room or your greenhouse and that airspace contact, that air in that leaf that's come through the stomata, that has to be at the right level to drive this system optimally. If the roots get too dry and we don't have enough water to cool the plant or to supply the water needs to maintain turgor, that rigidness in the plant will do things like shut the stomata down to prevent more water loss. If you shut the stomata down, now you're not bringing in CO2, you're not exhaling oxygen and water vapor, the plant can overheat. So we have to maintain root level hydration at the proper level by a number of different tools, and vapor pressure deficit is another aspect that has to be really carefully watched. Soil temperature, air temperature, all of these will affect this water potential gradient, and if we don't maintain the proper water potential gradient, the plant is not growing optimally, and we're seeing reduced yields and reduced quality. So we've covered a lot of science today. Hopefully we have looked at some things that will give you an idea of how complex the entire system is that you're trying to create an environment that is optimal for your plant, and that watering a plant isn't just going in occasionally and picking up the pot, and if it's a little dry, I'll give it some water. Whether you're out in a farm field or you're in a greenhouse or a grow operation, if you're going to grow optimally and you're going to have the system operating in a manner that's going to give you highest quality, highest yield, you have to maintain precision levels between this water potential gradient. In the next video, we'll actually look at how you determine what soil you are growing in, whether you're a field farmer and you're growing in sandy loam or silty loam or high clay loam or soilless grow medium. There is actually published data that's available out there that will tell us the exact levels of moisture needed for optimal production for the type of soil that you are growing in. If you buy a cheap water meter that you stick in the soil and says, yeah, okay, this is fine, it may be fine for a sandy loam, but it's going to be way off for a clay loam, or it's going to be way off for a silty loam. We have to use parameters specific to the soil that we're using. So we'll hold it there. Like I say, we've covered a lot. Your eyeballs may be twitching a tiny bit, but to me this stuff's all fascinating, and this knowledge is what's going to translate into either just a successful home garden or your success as a commercial grower, whatever it is that you're growing, because our job is to maintain this optimal environment for our plants to produce in their optimal way. And understanding movement of water through the soil, all of the different things like gravity, pressure, osmotic potential, soil matrix potential, maintaining that water gradient differential between the air. It's that important. It's why commercial operations have onboard scientists and highly experienced grow room operators. It's that important to your overall success. So we'll hold it there. Again, your eyes may be twitching a little bit, but hopefully we've cleared up some issues for you. The next video we do, we'll start looking at taking all of this knowledge from the last 3 videos and saying, okay, how do I measure my soil moisture levels so that I can precisely irrigate for the type of soil that I'm using and the type of crop that I'm growing. And it's actually going to be easier than some of the stuff that we've covered, because we've pretty much covered all of the hard stuff or the complicated stuff to this point. So, as always, be good to each other. Fall's coming, so maybe we can have a good fall. We didn't have much of a summer this year. I will see you soon with the final, excuse me, with the final video in this series. And as always, you need to know what your plants need to grow.